presentation for the afternoon session here in Lake Research and Monitoring is by Noland Michels. He's talking about the environmental effects on Age-0 walleye foraging and growth using an individual-based model. You're up.
Thank you.
Yep.
Well, thank you everyone for coming to the talk. I'm really excited to kind of share a little bit of the tail end of my PhD out at the University of Minnesota Duluth. So this is very much a Midwest-centered focused talk, but it does most likely have applications here in New York State as well. And so the big focus of this was walleye and kind of trying to help at least parse out some of the idea of some recruitment issues. There's a lot of recruitment issues in the Midwest. And it's highly variable, and kind of trying to understand those lake characteristics, and that variability is one of the big goals of this project. And so one of the big things surrounding walleye, especially in the Midwest, is this idea of thermal-optical habitat. So having ideal thermal and optical habitat that overlap kind of in the epibenthic region that basically helps support those individuals. And so the idea is, like we described right there, where you have a temperature range, you're going to have, there we go, that ideal temperature at the random zone of the lake. And then of course you're going to have that random optical habitat as well. Now that of course will change with water clarity as well as time of day and a whole host of other things. But this is the general idea of this model concept that Lester et al. Developed in 2004. And so with walleye physiology, typically the young of year typically prefer about 25 degrees Celsius as their optimal temperature, and then that usually drops to about 22 degrees Celsius for those larger adults. However, Chad Mellon et al. In Gretchen Hansen's lab at the University of Minnesota has found that basically thermal optical habitat affects smaller age fish more. So typically about less than 250 to 300 millimeters is where you're going to have the greatest effect of this thermal optical habitat on walleye growth and recruitment. And then up to 450 millimeters is kind of their medium range, which they said had some influence, and then for adults it didn't really have much influence at all. However, we might have this shift of where that temperature and optical environment might actually not overlap nearly as much, so finding out how that zone and what the actual influence is. This previous stuff is usually focused mostly on temperature effects, and very little has been done in the optical environment, and that's kind of where we want to play around a little bit more with this. And so our main question was, out of various habitat parameters, what has the greatest influence on the growth rate of young of year walleye? And so the first thing we wanted to focus on, of course, was temperature, most likely the bioenergetic component. The second being light intensity, really trying to hammer out, instead of just like the thermal habitat, thinking about the optical habitat as well, and how important that is to both foraging and growth. And then finally, of course, prey density, which has a direct correlation to amount of foraging and amount of bioenergetic, or how much energy, like the energy density in that ecosystem. And so to do this, we basically approach this with an individual-based model, which basically helps look at those dynamic changes throughout the ecosystem, while also measuring that throughout all the fish populations. However, that does rely on having appropriate available environmental data such as light and temperature, as well as estimates, decent estimates for population size, as well as local diets of those species. And so for this, with our environmental variables basically surrounding building this individual-based model, we focused on the structure of the lake. So our model lake was Lake Escanaba Escanaba Lake in Vilas County, Wisconsin, which was kind of a little bit preluded in one of the plenary talks. So here we're going to have a depth from basically 0 to 25 feet with this various structure mimicked here in this diagram. Temperatures, basically the lake stratifies around early May, and then from there stays stratified until usually about mid-October, and you're usually ranging about 24 degrees Celsius as your surface temperature, usually 22 degrees Celsius around kind of like the middle of the year. And then the other parts that we wanted to add into the model as well is thinking about the light, but not necessarily just time of day or water quality. We also wanted to try and factor in how much tannin concentration there would be. So in prior stuff that we did in the lab, we basically tested water clarity by using different tannic concentrations to understand what that kind of, what the extinction coefficient, the extinction coefficient, there we go, for Beer's law and stuff like that, to calculate that light at depth to actually understand what the optimal habitat those fish were actually experiencing. And so for this, we actually broke this up into basically 15-minute bins per day, or throughout the day, kind of specific light intensity, surface light intensity. And we then use Beer's law to actually calculate the light at depth for each specific zone that a fish is actually occupying. And so to estimate estimate foraging in this individual-based model, we use the foraging rate potential equation here that was developed by Mason and Patrick in 1993, which essentially is using the reaction distance of that fish, followed by the speed of the predator, the speed of the prey, and also the density of the prey at any given time. And so for this, we use some data that we collected in the lab prior to this, looking at the reaction distance of Age-0 walleye. And so here, down on our x-axis, we have our light intensity from 0 to 1,000 lux, as well as the conversion to photons per meter squared per second, mainly because that's a more biologically relevant unit than lux is when you're looking at fish vision, followed by reaction distance over here in centimeters on our y-axis. And so just to give you a little bit of idea, most of the foraging where you're going to have increased capture success and ingestion of your prey is happening around this 0.05 to 1 lux, which is actually approximately equivalent to about nautical to civil twilight. So really think about those really early and really late dawn dusk periods. Now in the model, of course, when you run it the first time, you realize all of your prey are gone, and that's because there's no satiation. So we had to add an satiation constant, and then we used that for some growth experiments that we did in the lab as well. And we found that basically about 3 prey every 48 hours is what was satiating our predators. So we added that into the model as well, followed by all of our bioenergetic components. So for this, we had yellow perch walleye system as the simplified system in Escanaba Lake. And so we had a bimodal yellow perch distribution, and then we had a random number generator to basically see where that pulled out of that bimodal distribution, compared that to the size selective date predation of these walleye, which grew over time as they grew. And then from there, we understood where it was and multiplied that by our temperature dependent scaling factor for our bioenergetics. So using fish bioenergetics 4 to create an individualized scaling factor to say, for example, at 22 degrees Celsius, 18% of that individual yellow perch's energy went into somatic growth for this walleye to get how much this walleye could actually grow. And so for this, after our foraging section, we needed to have movement. And so for our movement, especially with our walleye, we used the equation by Railsback et al. In 1999 and Thomas Robick et al. In 2014, which focused on these walleye actually evaluating the cells around them. So the 26 surrounding cells, they could evaluate and see where the highest prey density was, as well as scaling that to, are these within standard size ranges that they could actually consume, potentially, kind of rough estimates. And so from that, these individual walleye could then evaluate all the cells around them with how many prey there were and select for the higher prey densities around them. Not always selecting for always the highest prey density, but as long as it's basically above 3 if possible, they would go to any random spot there. However, earlier stuff from my master's, my PhD, I did a lot of prey of winds work. So this would be nothing without actually having at least some minor response from the prey. And so the prey are basically locked to their original cell that they start at, but then they can go within 1 cell of that. So basically they have 27 cells that they can run around in basically a giant cube. That's what their movement was limited to. However, they got to move after the predator moves, so just in case it's a random movement, but they may actually distribute out of that cell or stay in that cell. So. Next, we wanted to ground truth this IBM. So here, we have our growth in grams per gram per day of in-lab studies that we did under different light intensities. Here is 10 lux, 100 lux, and 750 lux, indicated by the shape, as well as the staining intensity, so thinking about a clear water system being green, 10 milligrams per liter of kind of like a medium tannic concentration being that kind of goldenrod, and the 20 milligrams per liter is what the highest staining intensity you'll see in some of those Minnesota, Wisconsin lakes. And so as you increase staining concentration, typically you have higher growth rates. As you increase clarity of the water, you're going to have typically decreased growth rates, unless they can easily find a structure or something like that to avoid in a real lake. So for here, we wanted to try and mimic this in our model. So we took our single light intensity at each day, and then we put that at the surface light intensity and kept that throughout the water column as we did in the tanks. We did this under a 14-10 day-night cycle, 18 degrees Celsius, add the minimum food. So basically here we just threw 20,000 prey, basically, just add the minimum food. and then tested this in each extinction coefficient to kind of get an idea of where those growth rates within the model lie, or where they lie compared to what we actually observed in the lab conditions. And what we saw, especially at lower light intensities, we saw pretty accurate growth rates throughout the system with a little bit of overestimation in our really low light intensities and a little bit of overestimation in our really high clear light intensities as well. But overall, ground truthing seemed to actually fit pretty well with our proposed, or our observed growth rates in the lab. And so our last part is we really want to try and conduct this habitat sensitivity analysis to really get this question of, is it temperature, is it light intensity, or is it prey density that's actually affecting these growth rates in these walleye? And so for this, we generated our nominal model, which took the prey densities, averaged over 2008 to 2014 from Escanaba Lake, and that was essentially 30 prey per acre was what the usual yellow perch densities were, of course with various fluctuations. The light extinction, like the light intensity and the extinction coefficient was approximately 3.5 per meter, so essentially that's how much is being pulled off as you go down in Beer's law using K. And then finally, our temperature-dependent bioenergetic factor was 0.18, or kind of 18% of that energy was being pulled off and allocated toward somatic growth increase. And so then, with the habitat sensitivity analysis, we take each individual factor and change that by plus or minus 20% to see how that individual factor will ultimately influence that variable that we're looking at, i.e. growth for us. And so with our results, here on our y-axis we have our percent change, as well as on the x-axis we have our nominal, as well as our low and high characteristics for each individual thing, including prey density, light intensity, and temperature. I'd just like to highlight right now, with temperature, our y-axes do have different scale bars, mainly because it's a much stronger influence, yet we wanted to still try and show prey density and light intensity with approximate conditions. And so out of this, prey density and temperature both had significant independent effects, mainly between the high and low conditions as well. But the big thing is, temperature was the most important, which probably isn't a surprise to most of us, being that is our bioenergetic component as well. In really cold water, they're able to support themselves with higher growth rates, but warmer water was more variable, but typically lower their growth rates. And so with that, thinking about this model and all this habitat sensitivity analysis, our models generate similar growth rates that we saw as our lab conditions as well. Temperature and prey density both have impact growth rates with significant effects in themselves. However, light intensity, although people think it's really important for this thermal optical habitat, didn't actually have a significant impact on growth rate, or at least in these models, compared to prey density or temperature. However, it could be a strong interaction with prey density. As we go back, you have a lot of variability with light intensity, kind of at that more stained light intensity. So it might be your localization to how close you are to your prey. Of course, reducing a little bit of that reaction distance. And so for a bunch of this work, I couldn't do this without all my collaborators. So both the Metzger and Brock labs, special attention to Quinn Smith from University of Wisconsin-Madison, as well as the various undergraduates that I had in our fish collection teams, including the Natural Resources Research Institute at UMD, and the Fond du Lac travel bans. I'll take any questions. Thank you. Yes?
I might have missed this when you were talking about it, but was your foraging success rate or reaction distance a function of the light intensity?
Yes, that is correct. So actually, when we showed the graph of reaction distance, that was actually plugged into each light intensity. So the reaction distance changed as light intensity changed. As well as the capture success rates, I forgot to mention, thank you for bringing that up. So at that kind of where we highlighted that red, that really twilight and silver twilight, their capture rates increased from about 9% unusual to about 22 to 23%.
And all those reaction distances were at 0 NTU?
Yes, that's right.
And do you think how that would change? Because none of these systems had 0 NTU, right?
Oh, absolutely, yes.
And so how would that impact?
Yeah, so Chelsea Neiman and Suzanne Gray have a paper that shows with, they showed turbidity, not necessarily with staining conditions. We tried staining conditions, it didn't work with our camera setups. But of course, it does reduce it. I think they found usually around anywhere from 25 to about 40% reduction in the reaction distance under highly like alkyl turbid situations. Yes?
I may have missed it, but the reaction distance in time due to optical visibility, did you work that both directions? Predator to see prey and prey to see predator?
So we saw the predator seeing prey. We didn't focus on the prey one for this one, but that is something that I always wanted to go back and play with that video. I just didn't have enough time to do. It's a great question.
I have two questions. Yeah.
Go ahead.
The first thing is about the light intensity. I was wondering why do you want to incorporate this factor into your model building? Could you, explain what's the role of the light to play in the